Thanks very much. So even more than normal when I'm giving a talk, I have a sense of imposter syndrome because as well as knowing nothing about statistics, I also don't really know very much about trials because I'm really right at the start of my career running clinical trials in dementia. But I wanted to echo what Pavel said at the beginning, which is I think one of the things I've really enjoyed so far is working with different people from across this campus. One of the things I wanted to add to that, which is I think there are sort of two groups of people with trials, people who come up with problems and people that come up with solutions. And so it's great to work with people who come up with solutions. And when you're stuck with something, instead of kind of going round and round in circles, you find someone that can kind of help you with a path forward. So thanks very much. I don't, as I've said, know very much about statistics, and so I'm going to assume a low level of knowledge about dementia. And apologies if some of this is very obvious to you, and you can kind of tune out and check your emails for a few slides. So I'm going to kind of talk about four things. I'm going to talk a little bit about dementia, why I think it's important that we do trials, and then I'm going to kind of pick three situations where I think innovative trials can be really helpful. I don't necessarily have a clear solution to those situations. You can kind of maybe come up with some yourself as we're going along. So we've heard a lot about COVID, which obviously was a very clear public health crisis a couple of years ago. But really in the background, dementia has crept up to be the leading cause of death in the UK. And a major cause of morbidity and mortality in the population. Even at the height of the COVID pandemic, a very large number of people were dying with or from dementia without COVID. So this is a slightly disingenuous slide, I admit, because it refers to sort of all-cause dementia against, say, just lung cancer rather than all-cause cancer, as you probably spotted. And so dementia is really an umbrella term. It's like diagnosing someone with cancer. It's not a very sophisticated diagnosis. Anyone in this room could probably make it in about 90% of the people that would come to see you in clinic. The thing says it's caused by different underlying brain illnesses, of which, after you get through initial screening tests, the vast majority are going to be caused by different neurodegenerative diseases. And really are sitting in the memory clinic across the road. The treatments that we have to offer people with dementia coming to our clinics have not changed for 20 years. We have a couple of evidence-based licensed treatments for Alzheimer's disease that have very small effect sizes, don't work very well. But for many other causes of dementia, we have nothing at all. And NICE, which you will all know about, recommends that we do not use any medication at all, due to the lack of benefit. So there's a big opportunity. I think one of the reasons that we are not making much progress in dementia and dementia trials is nihilism, which is that there is a perception that dementia affects people right at the end of their life. None of us are going to live forever. We all have to die of something. And people who have dementia have so many other medical problems that this is generally not a population where it is safe or feasible to run trials. But actually that's not really the case, because as we're getting better at treating other illnesses, and people are living longer, and they're living longer in better health, dementia is becoming more and more of a problem and really affecting people in the prime of their life. And so, actually, this is an accurate news article. It can kind of come along at any age, although obviously it's much rarer when we are much younger. But actually, even if you think about the sort of, in inverted commas, typical cohort of people with dementia, which is those who are over 65, these are people who have a long time left to live and a lot to offer society. So the UK pension age is now going to be, will probably be quite significantly higher than the kind of typical threshold when people start to get a very high risk of dementia. And without wanting to be too political, locally, one of the big arguments in this debate about the sort of mandatory retirement age in Cambridge was that people had still a large amount to offer at a very high level of kind of cognitive performance well into their late 60s. And so this is devastating when people in this stage of their life develop significant memory problems. You all know that the drugs in dementia have a very high failure rate going through clinical trials. And so I think there are lots of opportunities to how we can improve that. So I think one of the other things to say is we're really not short of targets. So as someone who wants to develop early phase clinical trials and do more trials in dementia, I think if you stopped all preclinical research, which is obviously still needed and that should continue, but if you stopped everything and just took the targets and compounds you already had available now, there would be enough work for me to do trials for the rest of my career. And this is targeting a whole range of different steps in the kind of pathophysiology of different neurodegenerative diseases. And as well as new compounds, we obviously can take advantage of repurposed compounds that we've already heard about, which may have either on-target or off-target effects from their original development that could potentially help people with dementia. But there is a significant lag in just the absolute number of clinical trials that we run in dementia. So this is chronic myeloid leukemia, which has a similar age-adjusted incidence to these rare forms of dementia together: frontotemporal dementia, progressive supranuclear palsy, corticobasal syndrome. And this is Alzheimer's disease. And these are the number of unique compounds tested in UK trials in a 10-year period. And I looked for a comparison for a single disease that I could put on the graph to compare with Alzheimer's disease, and there isn't really one because it's so much more common. And you can see that just the absolute number of trials we are running is far smaller than in other similar conditions. And I think the other reason that I like CML as an example, as many of you know much better than me, is it's a condition where clinical trials have transformed the treatment of this condition to something that was almost universally life-threatening with a very short prognosis, and there are now a number of excellent and highly effective treatments. So I'm going to sort of talk about three situations where I think innovative, adaptive, and even Bayesian trials, and I'm not going to talk about Bayesian statistics, can help people with dementia. So you've already heard a lot about platform trials, so I'm not going to kind of go through this again. The only thing I want to say is I think the people who've done the talks so far have been very modest about the effect that these trials have. So I think I'm right in saying, but please correct me because you probably all worked on this, the Stampede trial, which was one of the kind of initial large platform MAMS trials in prostate cancer. Before this trial started, there hadn't been any new treatments for metastatic prostate cancer for decades. Since the start of this trial, I think there have been 5 or 6 treatments, of which 4 of them have come just from this one trial. So I think it has the potential to be transformative in the clinical care of people with the condition included in the trial. So we are using this in neurodegenerative diseases. So MND SMART in motor neurone disease is the first platform study that really has got up and running in large numbers in the UK. I think one of the things that platform trials can be very successful in is including sites which are not sort of your conventional trial active sites if they're well designed. And this is something MND SMART has done very successfully. With initially 2 compounds compared to placebo, and then there have now been several further interim analyses, and we're now on to, I think, in testing for the 4th compound. All of the first 3 compounds have been shown to be ineffective. I think this does have challenges, though. I don't think it's the kind of solution to all our problems. And I think there are a number of really interesting issues that we still have to answer with these platform studies, which maybe people in the audience have the answer to. One of the problems is what you want to do if you want to add a treatment that is either delivered in a different way or monitored in a different way compared to all of your other treatments. All of the compounds that have been used in MND SMART and similar studies in neurodegenerative diseases have tested tablet therapies that require no additional monitoring. But actually that does restrict your potential options down quite significantly. And so I think it will be interesting to see how these studies adapt to include new interventions that may require that. And the other thing, which is I think has been talked about before, is how you choose the correct dose. So I think trazodone is an interesting example of this, because in Cambridge, over the road in the Medical Research Institute, trazodone has been identified in animal models as being highly effective for neurodegenerative diseases. And many of you will have seen these absolutely transformative data where you take mouse models of dementia, you give them trazodone at sort of human equivalent doses, and you can completely reverse the pathology that's seen in these animal models. And so there's a huge wealth of preclinical data behind all of these compounds. But these platform studies, at least in their current iteration, are not really testing the optimum dose. And so although you get a negative result at the dose that they're used, it's a bit unclear whether that was the dose that was really going to be most efficacious. The other thing I think that's really exciting is the ability to include more than one disease in the same trial. So the example that I'm going to use here is frontotemporal dementia and motor neurone disease, both of which you may or may not be familiar with. So motor neurone disease is an illness that affects the motor nerves. It causes weakness, here on the left in red. And frontotemporal dementia is a disease that affects the brain and causes change in personality and cognitive function. When I was at medical school, I was taught, which wasn't that long ago, I was taught that motor neurone disease only affected motor nerves, caused limb weakness, and didn't cause people to have any cognitive problems. But actually, we now realize that there is a significant overlap between these two conditions, even though they seem in the textbooks to be completely different. And some people with motor neurone disease, or ALS here, to use the kind of American terminology, have no cognitive function. And as you can think of from famous examples at this university, can be highly productive right the way through their devastation of their limb weakness. Other people have significant cognitive problems, about 30%, and about 10% of people with motor neurone disease, that cognitive impairment is sufficient that they have the full-blown clinical manifestation of frontotemporal dementia, and it works the other way around as well. And you might think that this is because neurologists don't really know what they're doing, and we haven't really kind of properly nailed the clinical phenotypes of these conditions, which I think is a reasonable critique always to have in the back of your mind. But actually, when people very generously donate their brain and their spinal cord after post-mortem, we can see under the microscope that they have exactly the same neuropathology. For reasons we don't understand, starts in different bits of the brain in different people. And you might think, well, actually, is this really the case? Is this really the same illness? Maybe there are subtle differences that we don't understand. But actually, we now realize about 10 years ago that people with the same genetic pathogenic variant, the same genetic mutation, can have the same either motor neurone disease or frontotemporal dementia. And there are families with C9 or F72 hexanucleotide expansions that have members of the family who have, some of whom have frontotemporal dementia, some of whom have motor neurone disease, and some of whom have both conditions together. We really don't understand why it is that some people get, what it is that predicts who gets which of these conditions, but they have the same underlying pathogenic variant, the same underlying pathology. And so it would make sense maybe to include both these conditions in the same trial. We did this a couple of years ago with a first-in-disease advanced therapy trial, a gene therapy targeting this C9 pathogenic expansion. So this was a sort of one of these really exciting studies where you bypass the first-in-healthy human step because you're delivering a gene therapy that is only going to be really indicated or ethically appropriate to use in people with the pathogenic mutation. And one of the things that this company very bravely did is include people either with motor neurone disease or frontotemporal dementia or both, with the primary outcome of safety and target engagement. And I'm not expecting you to read this, but this was the press release that they published just after we recruited another patient into the study, where they pulled the whole program, despite the compound being safe and engaging the target. And the reason that the that the trial was stopped was that they failed to show early evidence of clinical efficacy in this relatively small biotechnology company to justify moving such a complicated compound into later phase trials. The main kind of primary outcome used is this biomarker called neurofilament light that many of you will know about, which is the kind of current trend to use in neurodegenerative diseases as a measure of neuronal injury and death. But I think one of the things to really point out about this is that these two diseases are truly different. So this is plasma neurofilament light, and you can see that people with motor neuron disease have significantly higher values compared to those with frontotemporal dementia. So these are not the same illness. They are different, but they share something in common. And so we need to have trials that can handle this uncertainty, where there is similarities between diseases, but they're not exactly the same. And maybe we're able to model an overall effect across both diseases, but also to tease out differences between different subtypes of the illness. And I think this is something where forms of adaptive design that you've heard about earlier can be really helpful. The other thing we can do is kind of build on this a bit more, which is what we're going to try and do, which is treating a symptom common to many different neurodegenerative diseases. I'm going to show you data that I did in my PhD, and I'm very nervous that I think this is the first time I've actually shown this to people who actually know anything about statistics. So bear with me. So we looked at 2,500 people whose relatives completed a care questionnaire before their appointment. And what we were trying to do was use this to identify sort of latent behavioral and cognitive phenotypes that you're not really able to measure with a single question. And so we ran a principal components analysis. So this is just giving you one example of a component looking at behavioral disinhibition reflected by these questions here. And what you can see is these are 12 different neurodegenerative diseases, including Alzheimer's disease here in blue, frontotemporal dementia in red. But you can see actually that these symptoms are not restricted to a single diagnosis. So this is the kind of canonical phenotype of someone with frontotemporal dementia. But actually, not everyone with frontotemporal dementia has this clinical phenotype. Some people don't have it at all. And some people with other illnesses express it to a very high extent as well. So you might think if you want to do a clinical trial of someone with challenging behaviors, that what you should just do is have your population as people with a diagnostic label of behavioral variant frontotemporal dementia. This obviously has two problems. One is that you're not including everyone with the symptom you're trying to treat. But the other problem is it's sort of a practical, has less relevance really, because what you want to do with treatments is give them to people who you think might benefit from them. And so if you've got someone sitting in front of you in the clinic who doesn't have the symptom that you are trying to treat, then you're probably not going to want to give them the medication. And the other thing you're going to do is exclude people who have the symptom, but you haven't put in your clinical trial. So I think one of the things that's really exciting is the option of recruiting people across different diagnostic groups that have a common symptom to see if your medication is benefiting the widest possible group of people. When I started out thinking about this, my plan was just to group everyone together with a t-test or similar. But actually, the issue with that, as I quickly realized, is that these are not the same diseases. So if someone sits in front of you with progressive supranuclear palsy and semantic dementia, you will be able to tell them apart. People have very, very different problems. They tell you very different things about how this illness is affecting their life. But there is something about them that's in common. But they do have different underlying brain illnesses and different prognoses. So it's entirely plausible that people will not respond in the same way to the treatments. And so you need a way of being able to disentangle both an overall group effect and effect within your different indications. And Libby is going to tell you more about that tomorrow, so I'm not going to steal her thunder. The only other thing I want to talk about in terms of targeting symptoms is that, I used to think about trials as being either symptomatic or disease modifying. And I think particularly in neurodegeneration and maybe more widely, we're really trying to avoid that dichotomy because treating symptoms is very plausibly going to have a disease modifying effect. So the classic example of this is Parkinson's disease, where there is a selective loss, at least initially, of the dopamine producing cells in the brain. You can give people dopamine, replacement. But before we were able to do that 50, 60 years ago, Parkinson's disease had devastating prognosis and people died very quickly. When we gave people levodopa, that's not slowing the underlying disease, which continues to progress. So in that sense, it's a symptomatic treatment. But because it had such a dramatic effect, it had a dramatic effect on people's clinical outcomes. And Parkinson's disease has now been transformed, at least at a group level, to something which is not restricting how long people live for. And so you can see here what we're showing in this cohort of patients with similar underlying neurodegenerative diseases, that the degree to which you have this behavioral phenotype predicts your outcome more than just how long you live for, which actually most people don't really worry about too much, but how long you live independently at home with your family doing the things you want to do. And this is over and above a whole load of covariates, including the diagnostic groups. And however aggressively you try and analyze this and just restrict it to particular subtypes, this still comes out as a very strong effect. And so our hope and our question with the trial that we're doing is to see if we can move people from this group of severe behavioral impairment down into this group with less severe symptoms. We might be able to make a meaningful difference to their quality of life. So what would we use to try and to do this? What treatment would we use? I mean, one of the very nice things about doing clinical trials in neurodegenerative diseases, there really is true equipoise. So this was a study of a kind of survey of 50 people with 50 specialists who treat people with FTD. And you don't need to read this. This is just the list of medication people use. So really, there is a no clear consensus. Very happily, after we picked our compound, they came out with the suggestion that we'd already decided, which is to use citalopram. So I'm not going to talk to you about this in any more detail because Libby will go into the detail and knows much more about it. But we are hoping to start this trial later in the year. Funded by Race Against Dementia and Rose Trees. I met them at a seminar, and they told me that the aim of this program was to fund research they thought was so mad that no one else would do it. So I don't know, no one else would fund it. So I don't know if I should be reassured by that or not. But anyway, so it's really exciting. And I think one of the things that we can do with academic clinical trials is test ideas that maybe, and test ideas and methods that maybe industry are reluctant to use. Obviously, the failure rate in dementia trials is so high that I think when people have developed a novel compound, they're quite reluctant to take more risks about novel designs. And so one of the things that we can do, hopefully, is irrespective of the kind of effect on the primary outcome, is to show that this is a feasible, a practical and efficient way of running clinical trials. Just finally, in the last few minutes, I wanted to move a little bit beyond trial design to say there are other innovative things we are doing. So one of the big problems in treating dementia is that we have evolved a very efficient way of keeping things out of the brain. So it's quite hard to get your compounds inside the brain. And so this trial, also in frontotemporal dementia, is going to involve very innovative delivery where the patient has an operation which lasts 10 hours, and the operating room is there's an MRI scanner in the operating room, and over the very long operation, a neurosurgeon places under very careful guidance under the within the MRI scanner a very small tube into a key area of the brain called the thalamus, and injects very slowly a AAV vector containing the gene which is not functioning correctly in this cohort of patients. And in non-human preclinical models, this has a significant effect not only in the expression of this key protein, which there's a wealth of evidence causes this form of frontotemporal dementia, but also because of the way that the AAV vector is designed, spreads efficiently to other regions of the brain. This at the moment is open label, single arm, but I think it's really can be really interesting to see, assuming this is safe and effective, how they translate this into later phase trials, because this gene variant causes such a range of different clinical symptoms. And I think we may be coming back to some of these designs later in the program, the development program. The other thing is it's very difficult to have a placebo arm. So it's going to be interesting to see what comes out of that. And there are trials in Parkinson's disease where they do sham surgery, and just instead of drilling all the way through the skull, you have an operation, you have a general anesthetic, and they make just a small incision in the skull so that there's kind of to try and preserve blinding. And that obviously has significant ethical and practical problems. And then very finally, how am I doing for time? Quite well. So I think the other thing I have learned is that really you can kind of move this the other way around, because there are lots of ways that we can involve people with dementia in the design of trials. And I think one of the things that can be really helpful is actually pushing back maybe about some of the nihilism that I told you about at the beginning of the talk. And I think including the people with lived experience of the illnesses can be really helpful in really justifying that people are prepared to not everyone, obviously, but enough people are prepared to enter clinical trials that are testing novel treatments that maybe have more intensive schedules of assessments, and are willing to really commit significant amounts of their time very generously to try and kind of move progress forward to better treatments in the future. So thanks very much for listening. So I think hopefully I've persuaded you that we need to do more trials in dementia. Hopefully I've persuaded you or given you some information from the clinical perspective about where innovative designs can be helpful. There's certainly no shortage of people to take part in the studies, no shortage of things that we can test. And so I think we're really just sort of restricted in the number of clinical trials we can run. So thanks very much for listening, particularly to Pavel and Libby for all of the help that they've given me so far, and I think hopefully going to continue over the next few years, as well as all of the dementia team in Cambridge, particularly James Rowe, who leads the FDB group here. Thanks very much.